In this video, we will discuss about the in vivo results of AOH1996 on the orthopedic model of PDAC. So basically, this orthopedic model means that the tumor cells are directly injected into the mice model at the site where these tumors are naturally found to be occurring, and the experimental overview is shown in the panel A, where we can see that they took the mice model, that is ES1E SCID mice, and then inject 1 million KPC cells, that is pancreatic cancer cells, that has this KRAS G12D mutations and P53 R172H mutations. And after 2 to 3 weeks of this tumor cells to grow in the mice models and its confirmation using bioluminescence, they randomize this mice and inject half of the mice with AOH1996 at a concentration of 100 milligram per kg, and for the control cells they did not inject any mice models. And after 5 days they euthanized these mice and checked different parameters. First thing, they checked the mice weight, and they do not able to see any significant difference in the control mice and the AOH1996 mice. Then they check the tumor weight, and they can see that in the AOH1996 the tumor weight is significantly lower compared to the control case. And they also check the DNA damage cells in the tumors. So in the control case we see a very low signal for the DNA damage, which is stained by this gamma H2AX phosphorylated signals, and in the AOH1996 we can see more DNA damage signals, that is indicated by this gamma H2X phosphorylated in this pink color. And thereafter they quantified these signals. So in the control case we can see the positive gamma H2X populations is well below 50%, whereas in almost all the cases, or in all the cases that can be seen here. And in the case of this AOH1996 treated mice, they see gamma H2X positive populations is higher, and is significantly higher or not is done in the aggregated form where they did the nested t-test. And here we can see that the gamma H2X positive population is significantly higher compared to the control case, where the p-values for the nested t-test is less than 0.0001. Then once validating the results using this orthopedic implantation of pancreatic cancer cells, they also did the studies on the patient-derived xenograft, where the tumor cells from the patients has been taken who has undergone chemotherapy, and they did the studies in the control case where they do not inject AOH1996, and in the case where they inject AOH1996, and they are able to see that tumor size growth rate is significantly lower in the case of this AOH1996 compared to the case of control. And they did their studies using the ANOVA analysis, where p-value is less than 0.05. and they also wanted to check the survival, the impact of this AOH1996 on the survivals of the mice who are infected with these tumor cells. So they did their studies on the murine KPC injected mice, and here they are able to see that the survival rate for the mice which are injected with this AOH1996 is higher, significantly higher compared to the control case where they do not inject AOH1996, as we can see the p-value is less than 0.05 using this log rank test. Next, they wanted to check whether it also has any other impact. So they check the mice weight. So they do not see any significant changes in the mice weight when they compare it between the control and AOH1996. So all these things together indicates that AOH1996 has a role towards suppressing the tumor growth rate, and can even leads to the increased survival rate, or when the patients gets infected with the AOH1996 that has been shown here in the mice model. I hope you understood the results that are in vivo results that are presented here which shows that AOH1996 has some positive impact on the pancreatic cancer cells affected mice models. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.